Hello and welcome to Nikolai's Genetics Lessons, and today I have prepared two very easy multiple choice questions for you, and as usual I recommend you to stop video here, read the questions, answers, choose your correct answer, and when you would be ready you can run video again and you can compare your answers with my answers and explanations. And here is the first question: crossing over of parts of chromosomes. Answer A: has no effect on the genetic linkage. Answer B: usually decrease the number of genetic combinations in population. And answer C: can increase the number of genetic combinations in population. And imagine that we have two sister chromatids here, and here would be centromere, and we also have another pair of sister chromatids here, and centromere also would be here, and these two chromosomes would be homologous chromosomes. One can be from the father side, another from the mother side, and as you see we have here two chromosomes and four sister chromatids. So each chromosome at this stage would consist of two chromatids. And imagine that also we have here gene A, for example here dominant gene A, and recessive gene A on the other homologous chromosome. And we also would have here dominant gene B and recessive gene B on other chromosome. These two chromosomes can combine and make one tetrasome, and between gene A and B crossing over may happen in many different places. And as you see, if two genes, gene A and B, would be at the other end of the long arm, so here is a short arm, here is a long arm, such event can happen in many places. But if we move gene B, for example, here, very close to the gene A, as you see, number of possible crossing over would be limited. So what result we can expect from such crossing over? We can expect that one chromosome would be original, as we see here, with no recombination. Another one can look like this. So for example, here would be centromere and dominant gene A here, dominant gene B here, and on the recombinant chromosome we would have dominant gene A here and recessive gene B here. Another variant would be also recombinant chromosome, and we would see recessive gene A here and dominant gene B here, and the last variant would be original chromosome that we see here. And of course, because these two sister chromatids are genetically the same, we would have a small a gene here and small b here, or recessive allele A here and recessive allele B here. And on our picture we would see recessive allele A and recessive allele B here. So as you see, we have four different chromosomes as a result of this crossing over, and if gene A and B would be at the other ends of the long arm, a maximum variant of the crossing over would be, for example, if we take for 100% here, here it would represent 50%. So number of recombinants cannot exceed 50%. But if we move dominant gene B closer to gene A, as you see, number of possible recombinations would be less, and number of recombinants here also would decrease. Now we can answer our questions. So once again, crossing over of parts of chromosomes, answer A, has no effect on genetic linkage, and this is not true. And answer B, usually decrease the number of genetic combinations in population, and also this is not true, because crossing over the process that can increase the number of genetic combinations in population. But you also have to remember that if gene A and B would be very close together, just like one next to other, we may not see recombinations here at all. And we call such gene that these genes are linked. But any number less than 50% of recombinants here also means that two genes are linked. So that means that two genes are on the same arm of the chromosome. Now we can move to the second question. Crossing over resulting in the inheritance of altered chromosomes by children occurs, and as you see here is the three answers: during mitosis, during meiosis, and both of the above. And of course this process happens during meiosis, answer B, and it was very hard for me not to say this word while I was explaining what's going on this picture, because we can see such process only during meiosis, and meiosis happens only in sex cells or germline cells. And this does make sense because we don't need every cell in our body to be unique, but from the point of view of evolution, the more diverse our progeny would be, the more chances that some of them would survive, would be better fit for the environment, and meiosis process that create different recombinants, as you see on this picture, ideally serve this purpose. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.